everybody. So now let us continue our discussion on helicates. So in the last lecture we were looking at metal-based systems as supramolecular self-assembly methods. We looked at ladders, we looked at racks, and we saw how this specific composition of a set of ligands along with a metal ion can actually create a template which can actually lead to the formation of larger size species which are thermodynamically stable entities. So we did appreciate the principles of self-assembly in rigor in the last couple of lectures. Today we are going to discuss about another class of self-assembly processes involving helicates. In this case, what happens is that the coordination complex adopts a helical geometry to start with, and the molecular threads that gives rise to the formation of helical complexes are called helicands. So these are the ligand based backbone or the ligand based chemical entities that can give rise to the formation of helical complexes, and these ligands are called helicands, and the resulting complexes are called helicates. So to start with, let us look at one specific example of a simple helicand. So to start with, the ligand has got a backbone or a geometry or a conformation which already has the helical twist associated with it, and for example we can consider a simple helicand So we have this particular complex which can be formed when you have a metal ion N plus which interacts with these 4 nitrogen atoms, and so this is one binding domain and this is another binding domain. So it's a ditopic ligand, and we can put bulky groups R here, where R can be equal to methyl or hydrogen. And what has been observed that in the presence of metal ion in the +1 oxidation state, for example copper +1, it can form either 1:1 complex or it can form a 2:2 binuclear non-helical complex or helicate, and it can also form a double helical 2:2 complex which has the tetrahedral geometry at the metal center. So, and the binding domain, as you can see, has got different number of donor atoms. So the number of donor atoms here is 4, the denticity is 4, but the topicity is 2 because there are 2 distinct binding sites here, and these can create the necessary arrangement of the metal ions around the particular ligand. This particular ligand is referred to as quarter pyridine. So let us see how these look like. So you can have a 1:1 complex. For example, here in this case we can have a 1:1 copper ion and a quarter pyridine ligand which is forming this particular complex. So that is the first case. In the second case we can have and this is my metal ion sitting here, and this is another metal ion sitting here. So this is a binuclear 2:2 non-helical complex. So you see that this is an individual strand here, and you have the metal ion which can now form a tetrahedral complex with at every particular site. And then we can have the third one, which is referred to as the double helical arrangement. So we can have this, and we can have now this is coming from the bottom, so this will come from and this will again come from the bottom. So this is bottom top, and then this is top, this is bottom. So we have this kind of a encapsulation. So you can see this is my arms. So if one coordination is happening from this side, the other happens from the other side, and then on the other end we will have the opposite orientation. So top, bottom, and the other side will have the bottom and the top. This kind of an arrangement will take place. So we have now got this ligand backbone. This is the ligand backbone, and here we are going to have the metal ion which is from the tetrahedral complex. This is again a 2:2 complex, and this, but it is of a double helical nature. This is of a double helical nature. So each strand of the helix is important here, and this together constitutes the double helical structure. Now there are some properties of these helicates. Let us look at the properties of these helicates or helical structures. This is very important to appreciate the stereochemistry. Now the first thing is that the two chiral single helices, if we go back, so each one is a chiral helix. This is also chiral helix. Okay, the two chiral single helices of the same handedness, that is either left or right fit together to form a double helix. This is very important. So both these helices which are now forming this helicate like structure, both these chiral single helices are of the same handedness. So if say this is one hand and if I have the other hand like this, these are the mirror images of each other, then the opposite chirality helices cannot form the double helicate structure. Okay, because they will essentially, when you superimpose one on the other, they will essentially come like this. whereas what I need is this kind of an arrangement. So this kind of an arrangement can happen only when I rotate the same helical structure of a given chirality and then allow it to superimpose from the opposite direction. So we essentially have a rotation of this single chiral helix which can rotate, and then it can form the tetrahedral geometry or the double helical or double helicate structure. But if we have of the opposite chirality, then essentially these are non-superimposable mirror images. As you can see, they are going one on top of each other. They will not be able to form the double helical structure or the double helicate geometry. So these only when you have both the helices of the same chirality, same handedness, left or right, they fit together to form a double helix. So number 2, as it can be now concluded that single helices of opposite chirality cannot form a double helix. So this is what follows, that when we have the opposite chirality, they cannot form a double helix. However, the double helicates which we have now formed, they consist of a chiral helical screw. So the double helicates are themselves chiral, and once you have something which is chiral arrangement, you can now draw the non-superimposable mirror image of this double helicate. So these do exist as chiral entities. These do exist as chiral helices of opposite handedness. So the double helicates consist of a chiral helical screw which results in the occurrence as left-handed, that is M, and right-handed, that is P forms. This is according to the Cram-Ingold-Prelog nomenclature. So we have the CIP, the Cram-Ingold-Prelog nomenclature. According to that, we have now got the left-handed M minus and the right-handed P plus helices. So these are some of the most important properties of the helical structures, and another very important property is that both single and double helices can exist as enantiomeric pairs. So they can both exist as enantiomeric pairs, whether it's a single helix or whether it's a double helical structure. But unlike the conventional combination of two chiral fragments, the double helix does not exist as two distereoisomers because the heterochiral combination of two helices or two single helices does not give rise to a double helix. This is also another very important property of the helices, that although the single and the double helices can exist as enantiomeric pairs, and because the double helix is made up of two single chain helices of the same chirality, okay, they are of the same chirality. So we have got now two chiral fragments, and whenever we have got more than one chiral fragment in an organic molecule, we know that can lead to the existence of diastereoisomers. But unlike that of organic molecules, here the double helix does not exist as diastereoisomers, because the double helicate itself is formed from single helices of the same chirality. If they were of the opposite chirality, then they could have given rise to diastereoisomers. So the heterochiral combination of two single helices does not give rise to a double helix, and therefore there is no scope for existence as diastereoisomers, because what gives the double helical structure is the chirality of the helix, which is of the same type. Heterochiral combination does not give a double helix. So now when you have got a helical ligand, you can do some interesting electrochemistry with these metal complexes. Now given a metal ion which can form both an octahedral as well as tetrahedral complex. So now you can now have a metal ion which can exist different coordination environments, depending on the oxidation state of the metal ion. Okay, and depending upon the oxidation state of the metal ion, it will result in the formation of different kind of helices, and this information again is encoded in the system depending upon the coordination preferences of the metal ions. So let us take one specific ligand here now. And then we can have this one here. Okay, so now you can see this is having 6 nitrogen atoms. Now you can have two possibilities here. If you have got a copper ion, it has a tendency to form octahedral complexes. Then we need a coordination number of 6. Then there will be two distinct binding domains. This will be one particular binding domain and this will be another binding domain. So here the copper ion can now interact and it will form a 6+6 copper 2 plus complex. Or if you have other metal ions like iron 2 plus, cobalt 2 plus and cadmium 2 plus, they can form a 6+6. They can form a 6+6 double helicate structure. So we can have, and if we have a metal ion in the D10 system, for example, in the +1 oxidation state, it can form tetrahedral complexes. Then we will have 3 distinct binding domains and the topicity will be 3. It will be a tritopic ligand. In the case of octahedral, it will be a ditopic ligand. And then we can now have, because we are now going to have 4+4+4 copper +1 helical structure, then we can get a 4+4+4 are double stranded helix, and we can actually now do an oxidation of the copper 1 center to copper 2, and it will now change from a 4+4+4 to a 6+6 helicate, depending upon whether you have done an oxidation or reduction. So you can have the shuttling of the you can have the shuttling between these two states. So these are two different electronic states, one in which the copper ion exists in the tetrahedral arrangement. If you oxidize copper, it will now shuttle or change into the 6 plus 6 arrangement, and again when you reduce it and come back to copper plus 1, it will again bring it back to the 4 plus 4 plus 4 helicate structure. So this kind of a control can be taken place, can happen when you have metals in specific oxidation states, and essentially this can function as a nice electrochemical system where you can control the electronic motion and the nuclear motion as well, depending upon the oxidation state of the metal ion. So with this in mind, this is what we have to discuss about the metal complexes. Now towards the end of this particular lecture, I would like to take up a couple of case studies of self assembly processes in DNA, which is also called a molecule of life. This is also called a molecule of life. So here, what is DNA? This is responsible for encoding the genetic information that makes life possible. So this is what is the function of DNA, and what is characteristic about the DNA is that it is only the double helical structure that is the active form of DNA. So DNA is a very unique example where if you have two single strands of the DNA, and if you simply bring them close to each other and allow them to interact with each other via the formation of the hydrogen bonds between the ATGC pairs, it is not known to be the active form of DNA. So if you were to simply have two simple strands of DNA, you bring them close to each other and they are going to form these hydrogen bonds, but that is not going to be the active form. And what has been realized is actually that it is the helical twist of the DNA which further reinforces a pi-pi stacking interaction between the base pairs. So now we have got the 4 base pairs A, T, G and C, and it is a simple single strand DNA interacting with another single linear stranded DNA. It does not give rise to the active form. It is actually the double helical structure. And here we can see again that this particular information is contained within the system, and here we would like to see that how these ATGC hydrogen bonds exist. This is a very specific kind of hydrogen bonding, and we can draw the ATGC pairs here. So we can have NH here, and we can have this is the backbone. So we have the adenine, we have the thymine, and you can see that there is specific hydrogen bonding here, NHN and NHO hydrogen bonds. So there are 2 hydrogen bonds here, which again make it very specific between A and T. And when you go to the GC pairs now, you will see and we have So we can now have the so this is my backbone, and we can have now NH here. So we can now have the specific hydrogen bonds NHO here, NHN here, and NHO here. So we have this bond, we have NHN, and we have again another NHO. So this is between the guanine and the cytosine. So there are two NHO and there is one NHN hydrogen bond, and here there is a set of NHN and NHO hydrogen bonds. So you see this is a very specific hydrogen bonding pattern. And now once you have this hydrogen bonds between the AT and the GC pairs, let us look at these distances. The distances are slightly long. This is around 2.9, this is 2.8, this is 2.84, this is 2.91, and this is 2.84. Although these hydrogen bonds are quite long, but they are highly directional. So the directionality is what is very important when it comes to this particular hydrogen bonds here. So so this kind of specific hydrogen bonding formation is there. And then now once you have these hydrogen bonds, you have these ATGC pairs. As you see, they have got a large surface area and they are aromatic in nature. There is electron density. So now they can further have the helical twist where these actually co-facially stack with each other via pi-pai stacking interactions. So first you have the single strands coming close, forming the hydrogen bonds, then this forming the pi-pai stacking, giving the helical twist to the structures, and in this process it is actually trying to make the DNA very compact, go into a very compact and dense conformation or a dense configuration, such that the hydrophobic parts interact with each other, the hydrophilic parts also interact with each other, and this gives rise to a major groove area as well as a minor groove area in the double helical DNA structure. So the formation of the DNA helical structure is extremely important, but how is it that this DNA double helical structure is formed? Again, the formation of the DNA helical structure, it proceeds via a nucleation and growth mechanism. And you see, this is the interesting thing about nature. You have nucleation and growth processes, but then you also have self-assembly, which operates over long time scales as well as length scales, because you can now assemble small units and make larger units, so you can increase the size of the molecule, and these processes can take place over a finite period of time. So for example, let us consider one strand to start with. Let us consider A, G, T, A, C, G, and T. And then we have got another strand which is called T, G, A, T, T, G, C, and A. So the strand here has the base pairs which are complementary. So A we know is complementary with T, G is complementary with C, T is complementary with A with T, A with T, C with G, G with C, and T with A. And so we now have the first hydrogen bonding recognition, where we have the T hydrogen bonding with the A, and then we will have the G which will hydrogen bond with the C, then we will have C hydrogen bonding with the G. So in this case, this is first of all the nucleation step. Now in the nucleation step, as we have also studied in our initial lectures, it is a very important step, and it is a step where there is a decrease in the number of molecules. So now you are creating the necessary ordering, because the two single strands, which were actually quite conformationally flexible, now need to come into a particular orientation, and that decreases the entropy of the system. So initially there is a loss in the entropy of the system. There is an entropy loss associated with the process, but as the process of growth now takes place, as more and more hydrogen bonds, are formed, enthalpic considerations take over. And entropic penalty reduces, and that is the reason why now this double stranded DNA is now easily formed by this hydrogen bond association. So this is also referred to as a zip mechanism, where you actually have two single strands, and it starts forming the hydrogen bonds from the base, and then finally it forms into the helical, it forms into the double strand, and then it can now form the helical arrangement as well. So this nucleation and growth process actually facilitates formation of the DNA, and here we will have the propagation step. This is the propagation step. This was the nucleation step, where they come close to each other, start forming the hydrogen bonds, and then they propagate over the entire length of the DNA strand to form the double stranded DNA. So, these are this brings us to an end about the discussion of self-assembly processes, and in the next lecture we will now take up the next set of discussions on other applications of supramolecular systems. Thank you.